All right, I wanted to record a video walking through the steps that we can use to answer these 3-point mapping genetic questions. These questions tend to be amongst the most difficult for our genetics class overall. So I hope to break this down step by step so that we can make it a little bit easier as we move forward. So this will be one example. I hope to record an example walking through a second question later on in the week. So in order to do these 3-point mapping questions, we're going to follow some specific steps. We're going to take it one step at a time. We can't just jump to the last part of the question and try to answer it. We have to build our way up to that. So we're going to start by determining if the data we see supports linkage, that is, that the genes are all on the same chromosome. Then we're going to see if we can determine the parental allele combinations, that is, which alleles of the genes are grouped together on one chromosome, the paternal homolog, and which are grouped together on the other chromosome, the maternal homolog. Then we'll figure out the order of the genes on the chromosome. Basically, we're going to answer one simple question. Which of these 3 genes is in the middle? And then the last thing we can do, once we understand that they are linked, what the parental allele combinations were, and which gene's in the middle, we can then determine the distance between each gene pair, how far apart the genes are from one another. All right, so we're going to use this example to walk through this problem. So in this example, we're going to take some data from an experiment that this researcher Rollins Emerson did. Rollins Emerson investigated a few traits in maize or corn. He was looking at the seed color, green or yellow, whether they had rough or glossy leaves, and then whether they had normal fertility or variable fertility. We know that green is dominant over yellow, that rough leaves are dominant over glossy leaves, and that normal fertility is dominant over variable fertility. And we've defined the symbols that we're going to be using. Uppercase V versus lowercase v. Here we've got a 2-letter gene symbol for rough versus glossy, GL. So uppercase GL or lowercase GL. And then for fertility, we're again going to use a 2-letter symbol, uppercase VA or lowercase VA. All right, so Emerson crossed some pure breeding plants with the dominant phenotypes to pure breeding plants with the recessive phenotypes. Notice that 100% of the F1 offspring had the dominant phenotypes. That makes sense. And then he took those F1s and test crossed them to a plant homozygous recessive for all 3 traits. We'll talk about test crosses more, but anytime you do a test cross, you're going to cross to a plant that is homozygous recessive for every trait. So we told you that in the question. I'm just going to hide my head so you can see the rest of this. So we did the test cross and the progeny were observed generating the following results. I'm going to show you the results in a table in a couple slides. And so we're going to go stepwise through and answer a few questions. We're going to answer the questions on the last slide. Here they are again. Are these genes linked? That's going to be number 1. Then we're going to answer, what are the parental allele combinations? Number 2. Then we're going to figure out 3, what's the order of the genes? And then 4, we're going to determine the distance between the gene pairs. So again, this is our setup. We're going to try to answer these questions as we go here. So the first thing we have to do is just pick apart that question. We want to pull out every piece of pertinent information. So the first thing that was in there were the symbols for the genes and which was dominant and recessive. So I've made this little table here at the bottom. If I was trying to solve this question on an exam, I would write this exact information down, so I didn't lose track of which trait was dominant, which was recessive, and what symbols I was using for the traits. But the next thing that we're told in the question is that Emerson crossed pure breeding plants with the dominant phenotypes to pure breeding plants with the recessive phenotypes. This is the P generation. So we want to fill in here, what are the genotypes of these two plants, the pure breeding dominant and the pure breeding recessive. So we can refer back to our symbols. If they're pure breeding, we know that that means they are homozygous for every single trait. So if they're pure breeding for green seeds, rough leaves, and normal fertility, they're going to have everything from this dominant column. And again, they're homozygous. So I can put in this genotype, uppercase V, uppercase V, uppercase GL, uppercase GL, uppercase VA, uppercase VA. Homozygous dominant for all three traits. And we make a cross of this plant to another plant that is pure breeding for the recessive phenotype. So again, this one's going to be homozygous for all the traits, and this time it's the recessive traits. So we know the genotype of the other parent is this. Okay. So we've figured out what's going on with the P generation in this example we're thinking about. So next, we want to figure out what's going on with the F1 generation. In the setup, it told us that 100% of the F1 offspring showed the dominant phenotype. Okay, does that make sense? We can figure out from the genotypes of the parents what the genotype of the F1 offspring will be, right? One parent is always going to donate a dominant allele. The other parent is always going to donate a recessive allele. So we should get F1 offspring that are heterozygous at every location and express the dominant phenotype. All right, so now we know what's going on with that F1 organism. It's heterozygous and it has this genotype. So then our question, our setup tells us another piece of information. It says that we take those F1 organisms, we just figured out their genotype, and we test crossed them to a plant that is homozygous recessive for all three traits. Remember, a test cross means a very specific thing. A test cross specifically uses a homozygous recessive organism. So even if I didn't say that we did a test cross to a plant that is homozygous recessive, you should know that a test cross is always done to a homozygous recessive organism. So in doing our test cross, we can fill in the genotype of that other plant. It's homozygous recessive for every trait. So now we know what's going on with the F1 cross. And then we can get to our first question. Are the genes linked? Is there evidence for linkage of these genes? So at the very end of our setup, it said we did that test cross. Again, we've got that written down right here. We took the F1 heterozygotes and we're crossing them to these homozygous recessives in this test cross. And then it said the progeny were observed, generating the following results. And so here's a table representing the results that Emerson got out of this cross. So now we have to determine, are the genes linked or not? Are these genes on the same chromosome or are they on separate chromosomes? So the way we should do that is we have to generate some expected values if the genes were not linked. How often should I see each of these possible phenotype combinations if the genes were not linked? That would be to say, if each of the genes was, the outcome of each gene was an independent event. So how can we do that? Well, I would say we just have to figure out the expected frequency of each phenotypic outcome. So first we'll do that for the green versus yellow seed trait. Remember, green is dominant, yellow is recessive. We know that we're crossing our F1 that is heterozygous for this seed color to a homozygous recessive plant. So we can set up a Punnett square. We'll put the heterozygote on top, the homozygous recessive on the side, and then we can fill in the boxes. And we can see that half the time we should get green seeded plants, and half the time we should get yellow seeded plants. So the probability of green is 1/2, the probability of yellow is 1/2. Then we should do that for each of the other traits. So we can do that for leaf texture. Again, rough versus glossy. One organism is heterozygous, the other is homozygous recessive. So we put the heterozygote on top, the homozygous recessive on the side, we fill in the boxes, and again we see that we should get 1/2 rough and 1/2 glossy. Okay, so 1/2 rough, 1/2 glossy. Then we should think about the third and final trait, normal versus variable fertility. Again, our F1 is heterozygous. We're test crossing to a homozygous recessive plant. So we put one individual on top, the other on the side. We fill in the boxes, and we see that 1/2 of the time we should get normal fertility, and 1/2 of the time we should get variable fertility. Okay. So if we go back to this table, we can say, well, what's the probability of getting a yellow, rough, normal phenotype? Well, the probability of yellow is 1/2. The probability of rough is 1/2. The probability of normal fertility is 1/2. If they're independent events, the total probability would be 1/2 times 1/2 times 1/2, which would give us an expected frequency of 1/8 for that phenotypic class. Then we could ask the same question about yellow, glossy, normal. Again, yellow was 1/2, glossy was 1/2, normal was 1/2. This one's also should occur 1/8 of the time if these genes were assorting independently. That should be true for yellow, rough, and variable, yellow, glossy, and variable, green, rough, and normal, green, glossy, and normal, green, rough, and variable, as well as green, glossy, and variable. They should all occur 1/8 of the time. So we know we're looking at a total of 726 offspring. What is 1/8 of 726? Well, it's almost 91 of each. We should expect to see 91 of each phenotypic class if we counted these offspring. And I think right away you can see, well, this data, what we observed, doesn't really match. And the way we could prove that is by doing a chi-square test. We're not going to walk through that in this video, but we've done that in the past in this class. If you do the chi-square test, you should be able to prove that this outcome does not support independent assortment. And it tells you that indeed these genes are linked. So I'm going to stop this video here. That'll be part 1, where we addressed the layout of the question and whether or not the genes are linked. And then in the subsequent videos, we'll answer the remaining questions. So the first thing we did was, is there evidence for linkage? There sure is, because we didn't get the expected outcome from our cross. We got a lot more of some and a lot fewer of some other phenotypic classes. All right, so pick up with the next video when you're ready to go.